Hi everyone, and welcome to the third session of the online MOOCs, where we're learning AMR screening and theory. And this time we're learning about sensitivity testing, but also quite a bit about potentially setting up your own lab and sort of DIY AMR. As we've done every single time, reintroduce the course just in case. So online MOOCs are hosted on this website here. We're on Joggle, and that's where you can find out every sort of lecture and like the slides and a video as well. And so far we've learned about bacteriology and microbiology, and sort of the rise of AMR, and that was with me and Dr. Kurojisawa. And last week we learned about the techniques involved in the identification of microbes. So once you've sort of seen these microbes in the environment, sort of finding out what microbe that is. And then sort of this week, we're sort of learning about how do you test these microbes for susceptibility to various antibiotics. And also we're learning a bit about how to set up your own lab, because that's something that I'm quite passionate about. And so when you've attended these lectures, there's a certificate of attendance, and we're going to share a form in about 10 minutes, maybe 20 minutes. So you can sign up and say that you've attended. So yeah, here's the team. We might have more coming. John Moat, who was today's guest lecturer, he can't make it today. So he's the designer of a lot of these slides or ideas, and it was a pleasure to work with him and to refine my knowledge on antimicrobial susceptibility testing. We have Professor Courage, Linda, Gamily, Joy, Harry, and myself. And so today we're going to be learning about obtaining cultures, the quality control of strains, DOI techniques, AMR screening techniques, MIC testing, so the minimum inhibitory concentration, a little bit about antibiotic discs, the media to use, hints at better techniques. We're going to be looking at specifically the EUCAST guidance in AMR testing, because it's used so often. Spreading techniques, a tiny bit about robots and automation, and dangerous strains, and then that's it really. So I think to start, we have a little recap. So last week we looked at the identification of strains, and so we found strains from the environment or in cultures, and then we used a series of biochemical or genomic tests or molecular tests or assays to find out what strain that is. And this week we're looking at those strains, so now these are identified already, and using these identified and singular colonies cultures, we're going to see what they are resistant to, or even test new drugs. So to do this, I thought it would be nice to look at what like a minimum laboratory is for doing AMR screening and testing, so, or even as well indeed for the isolation of microbes and seeing what microbe it is to start with. So we need media, antibiotics, we need plates that can hold the bacteria on and hold the agar, and we need an autoclave, an incubator, and waste disposal. And of course we need people. And one thing that I thought was quite interesting is that a lot of equipment you don't have to buy new, and you can get from eBay, or you can get so there's lots of scientific equipment sort of secondhand schemes. So one in the UK, which I think is also international as well, it will cost quite a bit for shipping. It can reduce the price by like 95% from a new product. And so I was looking at this website earlier, and I found there was an unbelievable £2 million microscope that they were selling for like £6,000, and the same again for like much less expensive microscopes that would have been like a few £1,000s that are now a couple hundred, which is really, really interesting. So check it out, Uni Green Scheme. They've got quite a lot of equipment, as you can see. They've got 3,000 plus products. They've got secondhand centrifuges as well, which is quite interesting. Yeah, so that's kind of cool. So let's look at DIY solutions. So I guess to be in this sort of environment where you can work with microbes, you've got to make sure that you don't have contamination. So microbes that are floating in the air, they're very, very light. And so because they're floating in the air and going through the atmosphere, you have to have a way of creating sort of an area where they're taken out of the atmosphere or kept away from your plates. And so one way of doing this is the classic way of the Bunsen burner. So the Bunsen burner produces heat, and basically it just sucks up air around it and then pushes it outwards above. So there's like an umbrella almost around maybe a 30 centimeter area. You got to stick quite close to the Bunsen burner to make sure that you don't have any microbes accidentally falling onto your plates when you do this. But that's quite good. It's quite cheap as well. You can get them for Oh, I think maybe another panelist joined us. And they're just supplied with gas. So the main cost is the gas supply, and it's quite cheap, and you can use it on any table. So yeah, that's one way. And there's another way, which is for maybe if you want to work with a larger amount of plates. So for example, if you're a big lab, or if you're working with many, many plates, you're setting them into agar, you might want to have a big microflow cabinet. And these are much more expensive. So in the UK, they can be like £3,000 plus new, or even more. But I was just talking about that website, which you can get them a bit cheaper. So I would recommend getting them secondhand, because in the end, all they are is a filter and some air that is being pushed upwards off your plates, is pulling it through. So effectively, it doesn't go onto your agar. So it's not that complicated, and I would recommend probably getting it secondhand. Just wipe it up afterwards to make sure you don't get contamination. So another one which I find quite interesting is so when you are doing cultures, you have to autoclave it. So you have to sterilize before you set up a new culture. So for example, when you're making LB media, a lot of time, or a different type of media, a lot of the time you'll be starting off with products that aren't sterile, and so we'll have microbes in already. And the thing is, we have to get rid of those microbes in order to grow something new and pure in there. And the way of doing that normally is to use an autoclave, which brings up to 115 degrees C for 15 minutes. And it also pushes out all the existing air, because it might be that there's not an equal temperature throughout the sample. And so you get some parts of the sample that won't be 115 degrees C. So you've got to let a little bit of air out so the media itself can boil up and through the media, and so push out anything which hasn't been 115 degrees C. What's interesting about this, you don't have to use a very expensive autoclave, which can be up to a few £1,000s or a few £100s. In fact, you can even use a hot plate. Theoretically, you could even use a fire. Basically, what is actually required is to bring it up to a boiling point for more than 15 minutes, and to let the gas go through. And actually, all the time, because our autoclaves where I work are used quite often, I actually use a microwave, and there's a little protocol for using a microwave, which is much cheaper than an autoclave, for doing up to 500 mils at a time in two different shot bottles of 500 mils in glass bottles. And it works really well. I do lots of big cultures that way, actually. Yeah, and you can use water as well, as long as it gets up to 115 degrees. And again, you just have to boil the media. This can work with liquid agar as well. And then we've got incubators. So a lot of the time, people will think that to get their first laboratory together, they can have to spend lots of money on an incubator. But in fact, if you're not doing lots and lots of serious work, or if you need something below a certain temperature, you can just leave it on the side if it's an agar dish, and it will grow. It won't be as accurate in some cases. I'll talk about that in a bit. But you can actually do lots of microbe work without incubators. And so if, for example, all you had was a hot plate setup or a microwave and a Bunsen burner, in fact, you can do quite a lot of work, because you've got your sterile conditions, you've managed to create using this sort of umbrella of sterile area, as well as a sterile media starting dish, and that way you can do quite a lot of work actually, especially when you've got the flame to sterilize any metal equipment that you might use, for example, for moving discs across, or for using ethanol to sterilize a glass rod or glass inoculator, for example. Yeah, so another one is anaerobic work. So anaerobic work can require very expensive anaerobic hoods. And so anaerobic work, as we talked about before, anaerobes, there's a few different types of anaerobes. There's some that are sort of tolerant to micro amounts of aerobic material, so microaerobic, or proper aerobic. So there's almost, you have to have zero oxygen if possible. And they sell packs where you can reduce the oxygen concentration, and you can seal them in Tupperware boxes. Another one is the classic sort of the candle method, where you'll put your plates in, and then you'll light a candle, and then the flame itself will exhaust quite a lot of the oxygen inside, and so you can do quite a lot of anaerobic work without having to have a very expensive anaerobic hood. But in fact, you can use these. And also there's the copper sulfate and steel wool method. So you get a bag which is sealable, or plastic sheets that you can heat seal with steel, which with hot steel you might heat seal it, for example. You put the steel wool in with copper sulfate, and it absorbs all the oxygen in the environment. It rusts up, and by absorbing all the oxygen, you can do anaerobic work, and you can keep the plate in the same bag, which is kind of cool as well. And also this for imaging. So I mentioned before about how you can buy second-hand microscopes for much cheaper than you can buy first-hand. But also you can do DIY techniques. So you can actually buy open source 3D printed software or parts that you can just assemble and throw in an instruction manual. It takes quite a lot longer, but it can save a huge amount of money. So that's OpenLab, openlabware.net. There's a little link here in the slides later. And so today we're going to be talking about AMR screening techniques and sort of obtaining antibiotics. And there's two big factors in this if you're doing screening of antibiotics. One is sort of the disc-based method, and one's the media-based. So that's the MIC sort of method, where you can see the sort of difference in concentration of the bacteria depending on the amount of antibiotic you put in it. And so the advantage of the disc method is there's very little manual work that goes into it. So once you've made the plates, once you've got a disc and you just need to measure the radius of the disc, then you can measure multiple antibiotics at the same time, and there isn't a lot of pipetting. And so also the amount of materials in terms of like pipette tips is reduced. But there is one disadvantage, and that's that these discs can cost quite a lot of money. And also it's not quite as accurate as getting MICs like this. So it can cost quite a lot in media as well to create this many plates. But the main cost is actually the discs themselves. The advantages of media-based is that you can use liquid-based antibiotics, and you can also sort of provide sort of the MICs for various compounds that you might be wanting to test. It may be a new antibiotic. So if you've been looking for a natural antibiotic and you want to increase the amount of concentration, you can find a good MIC from that, and it's more, it's better data than you might have from a disc, which only has a certain sort of concentration gradient. You can see there's a small amount of inhibition here, but I think it really does depend what you want to do and your sort of style, I suppose. But in fact, you can make your own antibiotic discs. So if you get a paper disc, you can get sets of ready-made little discs, and you can soak that in so this is in water, but in fact you can soak it in your antibiotic or antimicrobial of choice, and you might be screening for that. So yeah, so there's a few ways of doing your own sort of DIY AMR screening, and or rather not DIY, but just low cost. And some of these are very sensible. and I do it as well. So obtaining cultures. So a lot of time when you're obtaining cultures, you can get them from a catalogue, so the EUCAST catalogue, or from some cases from hospitals. You can go to the environment, you can isolate them as we talked about in the last lecture. DIY, so if you know where a microbe might be, you might be able to, so for Staphylococcus, or you might be able to scrape it off your own skin and then do isolation of that. If you're not doing if you're just seeing whether it would work on that species, it would be a quicker way of getting it than if you were to ask an academic or get from a catalogue, which can cost quite a lot. So catalogues normally will cost money. Academics, it'll cost money for delivery, of course. But a lot of the time, if you ask academia, there's sort of an unwritten rule where if you've published something, you can ask these people that have published in that domain and they've got published work on it for the strains they used. And so you can measure them, and a lot of the time they'll say yes and they can send them over. So yeah, I've done that a couple of times and it's worked, and everyone's quite nice in the academia for that. So here are a few culture collections, and there's big catalogues where you can, what various strains you can get. This is especially good for quality control strains, or just if you're interested to see what your new antimicrobial, what strains it might affect, or if you've got a hypothesis of various antimicrobials having a synergy, if they're together, then that's an interesting way of doing it as well. So yeah, you can go back to this later on, and this also be available on the MOOC online. So EUCAST, which is what a lot of this this particular MOOC will be based on. We've got a few quality control strains, which sort of say perhaps the suitability of an antibiotic to certain types of bacteria. So we've got E. coli, Klebsiella, Pseudomonas, Staphylococcus aureus, and then Enterococcus. And they've got various different characteristics. I won't go through all that, but if you're interested in the various characteristics they have and why that's interesting as a quality control strain to see what sort of patterns they might produce. So for instance, we've got beta-lactamase producers, and if you want to test your particular antibiotic against a beta-lactamase producing organism, one which is either strong or weak, then you can see if your drug would be useful against it. So if we tested penicillin against it, it probably wouldn't be very good. But if you tested like a combination of maybe penicillin and your antibiotic breaker, which allows the penicillin not to be broken down, so a beta-lactamase inhibitor, then that's why these control strains exist for that kind of thing. Quite interesting. Yeah. So culture storage as well. So actually, a lot of the time people tend to use the -80, but you can actually use a -20. So we've got a little table here, and it is standard to use a -80. A lot of the time we're actually reducing that to -70 degrees C now to reduce sort of the environmental impact. A lot of incubators are now kept at -70. But if you use cryoprotectants and glycerol, you can actually use a standard freezer. So you don't have to spend as much money on using a -80 freezer. In fact, your strains will be good for 1 to 3 years. So if you're doing like a 2-year project and you wanted to use these strains, then in fact you can just reculture using the standard freezer and just cryoprotect this. So it's glycerol or even semi-skim milk. Yeah, and you can even do freeze dried as well. So if you happen to have a vacuum pump and a way of snap freezing your cells, so maybe some liquid nitrogen, which is a bit hard to get, or isopropanol and some dry ice, and dry ice is quite readily available from delivery companies, that if they send you something, then you can snap freeze to -70, and then you can use this snap freeze sample, put it into a freeze dryer, and then if it's been freeze dried, if it's in the vacuum, and also at -80, these can cost quite a bit more. You can actually have your sample stored for 15 plus years, and you can just keep that in the standard freezer. Also, even at 4 degrees C, as long as it's sealed and it doesn't require, there's no chance of any moisture getting in. And also when you're setting up your laboratory, you've got to be careful about waste disposal and safety. So especially if it's class 2, you should always talk with your sort of local authority or university on setting up risk assessments, cost forms, just ensuring that you're actually doing the right thing. A lot of the time there'll be a sort of panel which can say whether you're allowed to do something or not. And so for instance, if you're working with a class 2 organism, a lot of the time you'll be using something such as ChemGene, and ChemGene you can order online through an order system to wipe down any surfaces or areas where, or even tubes where your organism's been, and also ethanol, so 70% ethanol as well. As long as you're careful and you follow the COSH forms and it's been accepted by your local authority, you can do these kind of things. And in the end, most of your waste will be incinerated, as you can see. And occasionally it'll be alternatively treated if it's got, not just if it's clinical waste, but if it's a dangerous, if it's a hazard to incinerate, then there'll be other waste such as landfill or alternative treatment. So yeah. And then media to use, you can often find recipes online for various different media, so string, EUCAST. But when you do use a different media, so if you were to use sort of your own media rather than the media that's recommended by EUCAST, then you'll have to make sure that you use control strains and sort of calibrate your results to make sure that they're consistent. And also, so obtaining blood. So a lot of the time, especially if it's a fastidious organism, and that is an organism that requires very specific nutrients and can grow quite slowly, you'll need blood. And you can go to your butcher, and you also find horse blood, which is quite cheap. And sometimes the horses aren't killed; they just drain the blood regularly. And then once you've got the blood, you can defibrinate. So blood, it has fibrin in, and that causes it to clot. And so to defibrinate blood, you find the blood, you literally get the blood, and then you mix it inside a glass vial with either beads or some kind of agitating agent for less than 60 minutes to ensure that you don't lyse the cells as well, you don't have hemolysis. But what you'll get is on your paper, on your beads, or on mixer you're using, you'll get a big clot of blood, and you take the clot out, and what you're left is with the defibrinated blood that you can use for samples and for agar plates. Would anyone like to ask any questions before we move on to the next bit? Because the next bit we're going to be talking about sort of techniques, and this is more sort of the DIY aspect. You can ask in the chat if you'd like, and I'll answer. I can see one hand up, so I'm just going to let Abdul talk.
Hello.
Hi.
Yes, that was a great presentation over there, Chris. I just want to ask a question concerning the preservation of your media, temperatures that you can. And I want to ask, with respect to liquid media, now how most often in cryopreservation, there is this activity of osmosis. And so because of that, mostly storing products or storing genetic materials or biological materials that has high quantity of water usually has some osmotic interaction. And I don't know what advice you have to say with regard to storing liquid media. Thank you.
Thank you. So I guess I guess you mean liquid media with bacteria inside still?
Yes.
Yeah. I think these could be the mechanisms by which they do lose their viability. To be honest, I would always put cryoprotectants in, because what you'll get is the lysing of cells. In terms of osmotic stability, if you're using certain media, the media should already be suitable osmotic level to grow the bacteria, and hence the recipe. And it depends how you freeze it. If it's a snap freeze, then there'll be no time for any osmosis to occur. If you use like liquid nitrogen, or if you use isopropanol, dry ice technique, for example, there shouldn't be any time for that to occur. It'll be occurring over an incredibly long period of time, perhaps. And even actually, the freeze drying process, a lot of the time this actually occurs naturally. So normally we have a freeze drying process where you do it deliberately and you remove the liquid after you've frozen it by applying a vacuum. But in fact, this process is occurring always anyway, just at a very, very low level. And so if you kept something in the -20 for 10 years, actually it's kind of freeze dried, and that'll be partially allowed by the osmosis of things away from the ice crystals. But I honestly, I wouldn't really worry about these kinds of things. It's more of basically what you're saying. It translates the shelf life. If we say 1 to 3 years, that's probably these are the amount of time where you're going to get an instability in the crystals growing, freeze drying effect, and osmotic effect, destroying whatever's inside. I hope that's okay.
Thank you.
Yeah. And just fat. I'll let Blanche talk. Oops, sorry.
Oops. Thank you very much for the opportunity. That was a wonderful presentation. I think my question, are you able to get me, sir?
Sorry?
Are you able to get me?
Yes, yeah, I can get, I understand.
Okay, thank you. So my question is with regards to the blood that is used, especially when preparing blood egg and chocolate. I've seen, I think the main thing that that you highlighted is the horse. I mean the blood that is obtained from the horse. I mean, what other sources of blood that can be used for making blood culture in the laboratory? And also in terms of storage, because when you obtain blood from the horse, how do you store it to avoid contamination? Thank you.
Thank you. So in terms of this, I don't normally work with blood agar or blood culture, honestly. This was sort of John's expertise, who couldn't make it today. But I'll try and answer anyway. When I was researching and making the presentation, it looked a lot like you could use quite a lot of different sources of blood, but notably definitely sheep. So I noticed there was quite a lot of sheep blood for sale. I was looking around at that, and that looked okay. In terms of storing blood, I would store it how it recommends if you're buying it. If not, in the fridge, or in the -80, if it's sealed, could be what's necessary, or as soon as possible, using it if it's in high supply. If anyone if anyone has any ideas themselves and can give an answer, then please put your hand up. I'll just put the hands down, and you've got your own opinion. Please do talk.
Hi, Chris.
Just also Yusuf. Let Yusuf talk.
Okay, thank you for presenting, Chris. Like
He has been talking about the blood used for preparing blood agar. However, for us, we used to have hemoglobin if we don't have blood. We use hemoglobin. There is a powdered hemoglobin which is supplied, and we measure the concentration. We measure just grams by making 5% of the solution, and that's the one which we mix with the nutrient broth or Columbia agar base. Thank you.
Okay, thanks you. Thanks a lot. Really useful. Okay, I think we'll stop this discussion break and go on to the next part of the lecture. Now, a lot of you might have seen some of this before. I'm presenting what I've been given by an expert, and also videos, and also I've worked in this for quite a few years now myself. So I'm hoping that that this next bit will be just as interesting. So we're now looking at the EUCAST screening and focus on its use. So this guide was sort of made by John Moat, and he has some interesting insights as well about what you might find. You might find resistant colonies, for example, in your samples. Some of his own techniques he's used, and I talked to him for many hours through this, so this will be quite interesting. Now, if you're a novice, then something interesting to say before we continue is we've got two types of bacteria when we're talking about these tests. We've got fastidious bacteria, so these require sort of special nutritional requirements and conditions to grow, and then we've got non-fastidious, and normally you can find out what kind of bacteria that is through catalogs or through research itself. But we've got hemophilic fermenting anaerobes, and you've got like Haemophilus, Bacteroides, and then on the other side, non-fastidious, so we've got Enterobacteria, Pseudomonas. I was growing Pseudomonas yesterday. I tend to work with these normally. If you're working with fastidious, you'll tend to be using quite a lot of blood. So for testing the susceptibility of your samples, you can either use unsupplemented Mueller-Hinton broth, agar, for the non-fastidious, or you can even use LB. So as I talked before with how normally you would be using Mueller-Hinton, you can use your own broth as long as you've got a control there, and you've tested it a few times to ensure that media is capable of supporting the organism and it's to high enough density. And then we've also got Mueller-Hinton with 5% defibrinated horse blood. So when I said defibrinated before, that's when you're getting rid of the fibrin, and you'll do that by defibrinating, and that's by agitating it and then getting rid of the clots. And then you can use beta NAD, and then finally you use that for the fastidious organisms. And then so for non-fastidious, you can use many different types of agar, but Mueller-Hinton agar is fine. And then for fastidious, such as these listed here, we've got the 5% horse blood, and as Yusuf was saying, hemoglobin as well. You can use hemoglobin. If you say 5% hemoglobin, I can let you speak. I think you can talk if you like, Yusuf, if you've got comments on how you made your agar. Okay, maybe later. So I would tend to prepare your media to the manufacturer's instructions. So also when you're making agar, so when I said before about autoclaving your agar, you don't want to autoclave when you've got the blood there, unless you're making chocolate agar. So you have to wait until it's cooled until 42 to 45 degrees, so we don't have lysis those cells. Yeah, so make sure it's cooled. Pour the plates on a level surface, make a uniform depth, and then you'll find it's 4 to 5 millimeters, which is fine. And I tend to get to about 25 mils for a typical circular plate. Yeah, so test each batch using quality control. So sometimes you'll find that different batches of agar actually has a different color, and that will make it so sometimes it could be difficult to read inhibition zones, or if there's low concentrations or high concentrations of certain cations, for example. So you might have to do quality control of these. When you make plates, you tend to store at between 4 to 8 degrees. That's within like a cold room or a typical fridge. And I tend to seal the plates that I make, and they can last for quite a few weeks. But they will eventually start to get contamination or to dry out, and you'll start to thin, and then they're not usable anymore. So, yeah. And before you inoculate, it's always best to ensure they're at room temperature and that they're dry. And so we'll have a little look at a video a bit more about that in a second. And then for inoculation, when you grow your bacteria, you want to make sure that the ability is equivalent to 0.5 McFarland standards, which means absolutely nothing normally. But McFarland standard is kind of a mixture of cesium with other things that it used to be, or it can be now a suspension of agarose, which reflects like a certain look. And so here you can see this is a McFarland standard of 0.5, and you can see the turbidity of this, and then you've got water against it, and then we've got yeast, and you can sort of use this and visually look at the different McFarland standard versus your sample to see that it's sort of the right concentration. And you want to be this concentration to ensure that you haven't over inoculated your samples. And you can keep this McFarland standard just for looking visually, and you want to look at the rotation as well of your sample to make sure that it looks very, very similar. Yeah, and also you want to look for well isolated colonies. So if a lot of time you'll have big, lots of big streaks where the colonies are right next to each other, but if possible, try and use well isolated colonies if it's from a non-selected medium. Yeah, so when you're doing the inoculation, I think there's a video here that we can watch together, which is quite interesting. A very posh voice here. And this is by EUCAST, so the European Commission.
Use a sterile loop or a cotton swab to pick colonies from an overnight culture on non-selective media. Use several morphologically similar colonies to avoid an atypical variant. Suspend the colonies in saline and mix to an even turbidity. The density of the inoculum suspension should be equal to 0.5 McFarland standard. It is recommended that a photometric device is used to measure the density of the suspension. Alternatively, the density can be compared visually to an 0.5 McFarland turbidity standard.
Where they're looking at the similarities. That's the normal way.
If the density is too low, add more bacteria. If the density is too high, add more saline.
Right, that's the sort of the first half. That's where you prepare the inoculum, and then this is part 2.
Use it.
So then what you want to do with that inoculum, you want to inoculate the plates. So there's like a specific technique of doing this. I think we'll play the video so you can see what's happening. You don't have to use one of these. They're using a machine to spin the plate, but you can make your own quite easily. In
the EUCAST disc diffusion test is based on 2 media. Unsupplemented murohinton agar and murohinton agar with 5% mechanically defibrinated horse blood and 20 milligrams per liter of beta NAD. This medium is called MHF. Unsupplemented Murahinton agar is used for non-fastidious organisms such as Enterobacteriaceae, Pseudomonas, Staphylococci, and Enterococci. The Murahinton
we've been through this.
Agar or inside the lid.
Yeah, so
should be visible on the surface of the agar or inside the lid. This occurs more often with plates stored in plastic bags or sealed containers. If necessary, dry the plates prior to use.
The agar itself above it.
Plates can be dried either at room temperature 20 to 25 degrees Celsius overnight or at 35 degrees Celsius with the lid removed for 15 minutes. Use the McFarlane 0.5 bacterial suspension optimally within 15 minutes and always within 60 minutes of preparation. Plates can be inoculated either by swabbing in 3 directions or by using a plate rotator. Dip a sterile cotton swab into the suspension to avoid over-inoculation of gram-negative bacteria. Remove excess fluid by turning the swab against the inside of the tube. Spread the inoculum evenly over the entire agar surface, ensuring that there are no gaps between streaks.
Depends on if you're using gram-negative or gram-positive. If it's gram-positive, then it's fine it being very wet, the swab, and if it's gram-negative...
For gram-positive bacteria, do not turn the swab against the inside of the tube and take particular care to ensure that there are no gaps between streaks.
And you can just spin the disc. So they have an incredibly fancy plate spinner, which you might be able to get online for very cheap. But I save quite a lot of time just spinning it as myself. That's fine.
When inoculating plates without a rotator, swab evenly in 3 directions.
Okay, so you're going to make a sort of grid pattern. And that's it really. The next video we might learn a bit more. Yeah, so just then we looked at the swabbing 3 directions, creating a grid. Also, if you use a rotator, which you can have as mechanical as well. Yep. And then there's a little bit about storing antimicrobial discs. So if you've bought them, then store them according to the manufacturer's instructions. If you can, then make them very soon before, if you're doing a DRI version, make them very soon before you do the experiment. And then to prevent condensation, allow them to reach room temperature before opening the containers. I do this a lot if it's in the -20, for example. If it's a powder, a lot of the time it'll pull the moisture in from the air and get wet, and you'll get awful. So this is sort of a general rule, not just for discs, but for lots of things. If you can, leave your sample outside for as long as possible until it's got to room temperature, and then open it so the moisture isn't taken in. Especially true for dry powders. Yeah, so this is about the application of the antimicrobial discs. They do have these sort of disc setters, which are quite cool. But you also, there's a little video on this as well. Yeah. So after you've made this grid, then you're going to be applying the discs.
15 minutes of inoculating the agar plates. The required disc contents are listed in the EUCAST breakpoint and quality control tables available on the EUCAST website.
Really useful website.
Allow discs to reach room temperature before opening cartridges and containers used for disc storage. This is to prevent condensation, leading to rapid deterioration of some agents. Apply discs to the surface of the inoculated agar plate, either manually or by using a disc dispenser.
If you do it manually, it's best, obviously, to sterilize the
discs must be in close and firm contact with the agar surface and must not be moved once they have been applied. The number of discs on a plate should be limited to avoid overlapping of zones and interaction between agents that may prevent reliable measurement of zone diameters. Invert plates and make sure discs do not fall off the agar surface. Incubate within 15 minutes of disc application and keep stacks of plates as small as possible to avoid uneven heating.
In terms of the uneven heating, I don't think it actually matters that much. We tend to store ours up to, well, to fill the incubator effectively.
Antimicrobial discs should be applied within 15 minutes.
So yeah, that's sort of the application of the disc. That's after you've inoculated. A lot of you, I'm sure, will have done this many times before. Sorry, please bear with me. Yeah, so then we've got the incubation of the plates itself. So these plates, you can keep them at 37 because a lot of the time the incubator tends to overestimate the temperature it's at, and actually the best temperature for them to be growing at is 36. And we mentioned about different ways of the fastidious organisms. You can increase the CO2. There's certain chemical packs where you can have a certain percentage of CO2 as well. So when I talked about little pouches that you can open, you can get between 0.6% CO2 in those pouches if it's specifically for that. And also you can as long as you quality control it, you can see how much CO2 reduction you can get yourself, and see if you can get incubation of these fastidious organisms yourself as well. Yeah, and then they've got all the various different conditions. Normally it's just overnight for most cultures, unless it's mycobacteria, and then mycobacteria can take even a few weeks. And then some types of bacteria, rambling on a bit now, but some types of bacteria can take much longer. So for example, if it's a predatory bacteria with Del Vibrio, and I used to work with Del Vibrio, and although this isn't used in EUCAST testing, it can take 14 days for it to grow, because it actually requires a lawn of bacteria there on the surface already, and then on that lawn, the plaques that it creates will take a very long time to form when you're doing genetic work. And it is a bacteria, so it's quite interesting. And also you can, these incubation conditions, you can do it at other temperatures. It will cause abnormalities in the antibiotics. A lot of the antibiotics require a certain amount of stress on the bacteria, and there's no stress if it's a low temperature, or there's certain types of stress depending on the bacterial species. So you might get strange results, but you can do it at a different temperature as long as it's quality controlled by your control strain bacteria that I talked about before with the various characteristics they have. Yeah, and I've just got a list of all the different incubation conditions, such as with the CO2, if they're fastidious or not. And so one of the sort of take-home messages, try and, when you do the inoculation, do it within 15 minutes, and then apply the discs after the inoculation in 15 minutes. And then finally, if you do, so this is sort of trying to say, don't do too many plates at once, otherwise you won't be able to stick to the 15-minute rule. Then again, if you are quality controlling this, then that should be taken care of. So yeah, so this examination of plates after incubation, so we can have a look. So this is sort of what you might be getting. So we don't, so when we before mentioned about using the rotatory, the disc rotator, if you do it too quickly, then what you can get is you can get streaks that are like this, especially if it's gram-positive bacteria, and you can see that it makes the lines not very good. It's quite hard to read. It's not very well spread, so it's not like that. Don't do it as close together as you can if you're doing it via the patchwork method. They don't have a patchwork method on the inside, so I think they're slightly suggesting try and do it like this if you can, but the patchwork method is really very good. Just try not to have gaps, I suppose. Yeah. And then you got different reading zones as well. So reading zones, it says to do 30 centimeters by eye, and there's a few different sort of nuances in this as well. And there's a little video on that. But it's to do with, you can get hemolysis, and so there's 2 rings for hemolytic bacteria to read, and you want to read the one which isn't hemolysis, so let's have a little video as well on that.
Results from susceptibility testing are read after 16 to 20 hours of incubation, unless otherwise stated in the EUCAST disk diffusion manual. A correct inoculum and satisfactorily streaked plates should result in a confluent lawn of growth evenly distributed over the plates.
Sorry, it's restarted there. Okay, here we go.
If growth is semi-confluent or individual colonies can be seen, the inoculum is too light and the test should be repeated. Sewn edges should be read at the point of complete inhibition of growth as judged by the naked eye, with a plate held about 30 centimeters from the eye. Measure inhibition sewn diameters to the nearest millimeter with a ruler or a caliper. If an automated sewn reader is used, it must be calibrated to manual reading. When reading sewn diameters, plates should be illuminated with reflected light. Do not use transmitted light or a magnifying glass unless so stated in the manual.
What's quite interesting about
Read unsupplemented Muir-Hinton plates from the back of the plate against a dark background.
So in 2 weeks, Harry is going to be presenting his app and also data input sort of application online, where you can take the API results that we talked about last week, and then you can take the inhibition results that we're talking about this week, and you can put them together. And we're sort of the goal is we're sort of trying to create a map initially of Ghana strains of antimicrobial resistance. And so if you're interested in that, please message. I mean, we'll talk about it more in 2 weeks' time, but that could be something very interesting for you if you're interested in participating in some community science, creating a map of antimicrobial susceptibility around Ghana of various different strains. These are especially from hospitals and from samples that you might be working with, or if you're doing your own project.
Read Muir-Hinton plates supplemented with defibrinated horse blood and beta-NAD, so-called MHF plates, from the front of the plate with the lid removed. In case of double zones or distinct colonies within zones, check for purity and repeat the test if necessary.
So we're going to talk about this in a second, but it's with colonies that are within zones, if it isn't a pure sample, then that's probably good. That's actually a good sign, honestly. But if it is a pure sample and you get colonies within zones, this often hints at sub-resistant populations of your bacteria, and hints that if it measures, especially if you're testing for bacterial susceptibility, that it might be a very bad idea to give this antibiotic to these patients, or if you're trying to look for a new drug, this sub-resistant population will very soon cause actual proper full resistance, and so you very much want to avoid it. This is very characteristic. I'll talk about this in a second.
If cultures are pure, colonies within zones should be taken into account when measuring the diameter. If zone edges are fuzzy, follow the standard instructions, namely, hold the plate about 30 centimeters from the eye and estimate the zone edge. Do not hold the plate up to the light and do not use a magnifying glass. For Proteus species, ignore swarming when reading inhibition zone diameters.
Yeah.
For hemolytic organisms, read inhibition of growth and not inhibition of hemolysis.
So hemolysis,
Beta hemolysis is usually free from growth, whereas there is often growth within areas of alpha hemolysis. To better differentiate between hemolysis and growth, tilt the plate back and forth and measure the zone diameter. Several examples of reading of inhibition zone diameter.
Yeah, so there's more examples on the EUCAST guide. I really recommend it if you haven't read it before, or if you haven't read it in a long time. It's very useful for finding techniques and for quality control. Yeah, this is just going over what you said before. So look for distinct colonies, double zones, especially hemolysis. Measures the nearest millimeter. Hold it 45 degrees with the black background. Very useful. And then we've got a sort of a table of different exceptions that you might have. So this is more for reference afterwards if you're going back to these lectures. Ignore isolated colonies within inhibition zone. I don't really like this kind of ignore statements, but with swarming, certainly you can. There's exceptions here that might find interesting. Yes, so now we've got sub-resistant populations, and this is an MRIC plate, I think, and these sub-resistant populations are the worst. So if for example you're recommended what antibiotics is this bacteria resistant to, then you can see that it's resistant to vancomycin there, the VA, and we've got SIP here, but it looks like honestly it is resistant to some of these antibiotics as well. And so in fact, it's better to test against furthermore antibiotics before going on to these, because what you might end up with is you might end up with a latent infection, which increases the resistance of this subpopulation. And so this would be what takes off the entire plate. And also this isn't a very good inoculation because you can see the individual colonies. So we've got a little storage video here as well. Storage of media and discs.
In-house prepared Muir-Hinton agar plates are stored at 4 to 8 centigrades, and the shelf life should be determined as part of the laboratory quality assurance program. Commercially prepared plates should be...
So by the quality assurance program, I suppose it means if your plates, you just have a cutoff. So make sure that if you've decided that your plates are good for 2 weeks, you have them for 2 weeks, and then you don't just keep on storing some of them for longer, but you sort of get rid of them at a certain point. Yeah, and so everything's kept at the same sort of conditions.
Make sure that agar plates have reached the lid. For agar plate, dry plates either at 20 to 25 centigrades overnight to moisture. Incorrect storage and handling of discs may result in loss of potency and reduced inhibition zone diameters.
So I mean, one of the if you're storing antibiotics, make sure storing them as proposed by either your collaborator or as on the instructions. I mean, it could be that they've degraded. Like there are some very so ampicillin, for example, degrades over time. We always keep it in the -20. And also different antibiotics will have different resuspensions as well. So some might be in ethanol, for example. A lot of people already know that. I don't think there's anything that useful in this video, but it does exist, and I recommend it and put in the presentation for that reason. So, okay, so there's a few interesting things. So when you're doing disc diffusion, you can get antagonism. So maybe one antibiotic actually allows inhibition of the other, or synergism. So here we've got resistant. So your bacteria is resistant to the antibiotic. Sensitivity. What's an interesting one? We've got resistant mutants. So when I said about subpopulations being resistant, these are very bad examples in terms of inoculation density. But in terms of resistant populations, that's quite important. Synergism. So synergism, if you're doing 2 antibiotic discs and they're very close together, what you might find is that the disc, actually they have a much larger area than if it was just alone, which is quite interesting, and might be something to note down if you do happen to study a bacteria and you notice that on the plates 2 antibiotics work very well. Maybe it would make sense to use both antibiotics at the same time, and that's something that's being used. It's to combat AMR now is to use multiple antibiotics, but what you can get is you can have the exact opposite, where in fact you get resistance in some bacteria caused by the existence of another antibiotic. So here you can see that the zone of inhibition for this antibiotic would have gone all the way out here, but because there's this other antibiotic, it actually makes it susceptible. And what you can have is you can have an antibiotic which actually sort of alarms the bacteria to say, hey, look, there's an antibiotic here. And so the pumps sometimes, or the resistant mechanisms which can allow these bacteria to become resistant or to stay resistant are turned on, and then that can make it more resistant to other types of antibiotics. So there's definitely two different, very different forms of antibiotic interaction here. We'll talk about tilting zones. Yes, and we've got some more control mechanisms. So before we talked about beta-lactamase, but we've got various different culture collection of Staphylococcus aureus, and so if you want to look at a specific antibiotic resistance mechanism, and if your antibiotic is still useful against that, you can use different control strains. Before. So you can do control tests to make sure that your samples are still good over a long period of time as well. So if you've been using a control strain for a few weeks, then you have to make sure you've got a consistent log of how often you're checking your control strains to see whether they're resistant, see if they're behaving the same, test them for different antibiotics, make sure that they're not accidentally introducing new mutations or anything from the environment. Yeah, and you can monitor the results to see if it's out of range. So if you plot these results over time, you might find that sometimes you'll just suddenly get huge or very, very small zone diameters. So you can do repeat tests and see if it's similar. And if it's out of range for 2 times in a row, then you should report this and you should investigate, because it might be that you've got accidentally have a new antibiotic resistance problem. And I've talked about storage before, but you can subculture your strains as well. So, one of the ways that you might keep bacteria is not just by storing in the freezer, but is by subculturing. So it's always good to have a -70 or -20 strain in beads of your bacteria and kept in glycerol, so you've always got a backup. But a lot of the time your lab will be using quality control strains constantly, and so you can take a small sample, especially from a plate, and you can use that plate sample then to inoculate a new one of your quality control strains each week, and then during that week you can then take the broth or you can culture that bacteria again the next day, grow up again, and then again and again for up to 6 days, and then so in that way you've got your long-term stock, which is stable. You've got your plate for taking new colonies or for setting up a new culture, and then for during the week when you're doing all your work, you've got your quality control strains ready for inoculation. Yeah. So we've got various different sorts of error, pH supplements, expiry dates, keeping to these rules, the reading conditions, the reading edges, and then the discs. So sometimes they weren't stored properly, sometimes there's too many on the plate, sometimes you use the wrong QC strain accidentally, so you might want to check that your strain's actually correct again via some of the methods last week, and sometimes the age of the culture. So if you've been subculturing for a long time, actually there's quite an interesting story about this. So Del Vibrio, which is a strain that I used to work on, the major strain that is used is the HD 100 strain, which is sort of so it's not rod shaped, it's comma shaped. It's got a comma shape, and that's what's in the wild type as well. And a long time ago, I think in the 70s, someone found or started using the 109J strain, and the 109J strain was the same in every single way, apart from one sort of almost imperceptible difference, which is there was very small bacteria, so it's hard to tell, especially when culturing, is that they were now rod shaped, or just they had no comma to them. And they found out that actually the reason why was because they've been subculturing for so long. They had a mutant, and the mutant had one difference in one gene, and the gene happened to be one of the peptidoglycan or modification genes, which change the shape of the bacteria. So now it's a rod. And so actually using accidental mutation long ago, they found out the reason why the bacteria became comma shaped. But it's not. But this is very, very obvious. You don't tend to find a mechanism or mutation which is so obvious in bacteria. If you subculture for a very long time, you'll start to get mutations which will be caused by the fact that you're using these bacteria in certain conditions, and they select for those conditions, and they don't have selection anymore. So they just rack up the mutations, and in fact, they might no longer be susceptible to certain antibiotics or gain susceptibility and stuff like that. So it's important to make sure that your quality control of these control organisms is good, so you don't lose the accuracy of your experiments and don't find the wrong result. And then we've got MIC testing. So before, everything we've been doing is talking about antibiotic discs, but in fact we've also got MIC testing. So that's minimum inhibitory concentration, and you can use a combination of different drugs, I suppose, like also you can with antibiotic discs, but you can actually reduce or increase the amount of antibiotic in tandem with your bacteria, and sort of create a 96 well plate of increased concentration of different strains and compare them against one another for turbidity, or to see if you get lysis or growth of your bacteria. So there's a little video on this which explains it quite well, and you can see how they're doing it as well.
MIC stands for minimum inhibitory concentration. MIC can be performed by different methods, including broad dilution, agar dilution, and E-test method. We have already discussed E-test method can easily be performed in a clinical laboratory. Antibiotic solution can be prepared. P stands for potency of antibiotic powder in microgram per milligram. To achieve
So we'll go on to the actual
antibiotic solution should be prepared at a concentration 10 times the highest concentration to be tested.
Yeah, so what we do is you do a sort of serial dilution of your sample. So you start at the 10 times the maximum concentration, and then you reduce that with a set of sort of ready-made. You reduce that by having it 10 times, and then into your cultures with no antibiotics which haven't been inoculated yet, you then take 1/10 of that and put it into the next one, then 1/10 and 1/10 again. You get like a serial dilution where you remove, you reduce the concentration by a factor of 10 each time. That way, it's sort of serial dilution series. You can change the accuracy as well. So it's instead 2 times dilution, you can get very accurate MICs that way as well. But it normally tends to be 10 times. And we're using lots of pet tips for this. Let's get on to the actual process itself. So once you've grown a culture of your bacteria in media,
the range of antibiotic dilutions in the microtiter plate should be 2-fold higher than the actual working concentration.
So you can see here they're doing 16, 8, 4. So they suggest that they're doing a 2 times dilution each time. So they're taking half of previous and then putting it the next to reduce the concentration. So we'll have, imagine if it was a 200 microliter maximum, which is the plate, they're probably working with 100 microliters of sample. And so they'll be doing 50 microliters of that, taking to the next one. And in that next one, there'll be media ready, maybe 50 microliters, and they'll take 50 microliters of that, put it to the next one, and in that way you get this dilution series. And then they tend to use multi-channel pipette, so we'll just have a little look at that. Let's get to it. Yeah. So the technique where what we tend to do is you tend to use sort of, actually a lot of time I use agar dishes for this, because these do actually fit with agar dishes, and within either 30 centimeters, 20 centimeters of your Bunsen burner, or in the microflow hood, you can do this, and you can put your media there ready, and you can take also the antibiotic that you've prepared, and you can put that in the plate ready as well. So you can just pull it with your multi-channel pipette here, and then you can just go along, and then when you're ready, you can then read the results. So just watch it. This
mix thoroughly. Serially dilute the cation-adjusted Mueller-Hinton broth and antibiotic solution in subsequent wells till 11A and discard 100 microliter from this well.
Yeah, and then you can have your various different strains as well. So you might be quality control strains in the first few wells, and then your strain that you're interested in the bottom few, or you might be checking your strain for various inhibition concentrations, different antibiotics. So yeah, so when you put it in the incubator overnight, 37, you can then take it out the next day, and you can read the results. And you can see here that when you reduce the E. coli, Pseudomonas aeruginosa, it's a strain of E. coli as well. If you reduce the concentration of the antibiotic, which is here, this is the antibiotic concentration at the maximum and reducing. You can see the quality control here. This is where the bacteria have grown to their fullest, and you can see the MIC. So you take the MIC where you've got no growth, and also like half growth. You can note it down as you'd like, but as long as you keep it sort of to controlled standards of no growth. Here you've got 1, so there's no growth here, no growth here, and that's where you take the MIC. So that's the minimum inhibitory concentration of the antibiotic. So they're testing strain 1 here against the antibiotic they're looking at. And then you've got your quality control strains as well. So it looks like E. coli had growth until here-ish, and then compare that with the test strain, where actually it's more resistant, and there'll be some strains which are far less resistant. And then you might use a different sample here. So this another antibiotic they're testing, and you can see that it's much stronger effect. And so there's growth at very, very low concentrations, but then no longer at higher concentrations at all.
Sterility control and antibiotic control should not have any growth. Organism, MIC falls within the recommended range. If control wells, sterility controls are open.
So also you've got to make sure that the organism itself it falls within the recommended range. So if you find that for some reason your control organism has the antibiotic, or if it's not a new antibiotic and it's known and it's not within the expected range, then you know something's up, and you should repeat the result. You should repeat the results anyway. Yeah. So let's go to the next slide. I think you might have accidentally pressed something there. Sorry. Close this. Everyone. And it's quite interesting the social media companies closing yesterday. It's the internet, but it's not that today. It's my computer being very, very slow. So whilst that's going on, I might as well talk about the next slide, which I'll just stop the share if it's possible. The next slide, which is dangerous strains. So when you're working with a new strain or a dangerous strain, it's basically the rule is just treat all your strains with extreme caution. Follow the risk assessment, and also there's a high likelihood that someone you've worked with has worked with it before, so they might have worked with the strain and have quite a lot of experience with it. Try and just learn as much as you can. Don't just do something without having thought through the risks involved, through how you get rid of the waste. So if you don't respect the strains, or if someone in the lab doesn't respect the strains, then be as strict as you possibly can with them, because they'll hurt someone. And that's the advice from John, and also from myself, and also the horror stories have gone on. So people who work with plague have died. People who work with lots of different things have become ill, majorly ill, because they've contaminated themselves with it. I think, unfortunately, my computer has crashed quite a bit, in terms of the graphics display. I might let Gamily take control if that's the case. But otherwise, we talked about the different DIY methods that you can use to do antimicrobial resistance. We looked at MIC testing and antibiotic disc testing and the quality control that is involved with both. And we looked at the different media, and I hope it's been useful. So yeah, and I think that filled in the full 1 hour and a half as well. So brilliant. I am just going to close various windows as we talk. Hi, can you hear me again now? Sorry about that.
Hi, Chris.
Hi, sorry about that.
Yeah, I stepped out for a moment and everything seemed to be very quiet. And because I don't have access to how do you call it, sign-in at the moment. I was getting a bit confused, but I couldn't reach Harry as well. I was almost about to log in myself just to sort it out.
Okay. Sorry about that, everyone. I hope. When did it cut off? Do you know?
I stepped out, but I believe it wasn't too long ago.
Okay. I think we just finished the MIC testing. Is that right?
Yeah. So it was roughly around 11:23. So 4 minutes ago. Yeah.
Oh, 4 minutes ago.
Yeah.
Oh, okay, brilliant. Okay, it's not too bad. In that case.
Okay. Okay. I'm working.
So the link. Has everyone managed to use the attendance link? Let's put this, everyone.
I've shared it multiple times.
Okay.
And most people seem to have filled it, but there are still 1 or 2 people asking for it. Please, in the future, just to make things easier for all of us, also try scrolling up a little bit to find the link, because we shared it multiple times.
Sorry, everyone, that I cut out there. I hope you found the talk interesting. I was just talking about dangerous strains, and in the last couple of minutes, and how we should respect them. So we had a look at the DIY methods of AMR that you could potentially use, where you might find equipment. We looked at the disadvantages and advantages of using MIC versus disc screening method for antimicrobial resistance, and we had a look at the quality control. We looked at the EUCAST videos and techniques that they recommend and that we use, and I hope it's been useful. Next week we're talking about the AMR stewardship. So what sort of techniques that the community or the government might have to use, or farmers might have to use, to reduce the amount of antimicrobial resistance that's getting into the environment and back into our lives and hospitals, and to reduce the chances of sort of an apocalypse caused by antimicrobial resistance, which we are approaching if we're not careful. So that will be very interesting, and that will be held by Gamli. I'm just gonna. Has anyone got any questions? Please raise your hand. If I missed anything, please
Chris?
ask as well. Yeah.
Chris, can you hear me?
Yeah, I can hear you.
Yeah, there's a question in the Q&A section. There's a question from Amis, and he's asking, I have seen that the advised method to inoculate the Muller-Hinton plates for antibiotics testing is through swabbing uniformly on the surface. My question is that, can I measure an inoculum from a fresh liquid broth and spread it by swirling the plate?
So
I don't know if you saw that question.
By swirling the plate, do you mean inoculate the plate by spinning, whilst also?
Maybe we can have Hamisi, if he's around, to ask the question, just to clarify that.
So I think you can talk, Menezi. Why is horseblood commonly used? Horseblood's used because it's just so available. See.
He's not available.
Okay, that's okay. Is there any more questions? Or any advice from, if you've got your own experience? Anything you'd like to say on how you do AMR testing? If you have any techniques that you might want to share?
I have a question.
Okay, cool.
Let me see if I can turn on my camera, just because I'm able to do that. Hi. So yeah, that was a really interesting presentation, and thank you for not just talking about AMR sensitivity testing, but also showing do-it-yourself approaches and low-cost approaches that can be used. And so my question is around aseptic techniques and quality control with a low-cost approach. has there been any research done on some of the methods that you've shared, showing that we can get maybe lab standard, high quality results from these approaches? And do you have any tips on, aseptic techniques when you are using this type of DIY approaches?
So in terms of whether there's any published research to say if the technique itself is as good, there isn't really. There isn't. I mean, to be honest, there is on the incubators or on the autoclaving of media that has been done, and there is research on it. I think I shared one of the papers that looks at actually how if you microwave your culture, it's somehow better than if you. But in terms of normally, actually, so many people use DIY techniques that it's you just don't put it in the methods. So you say they were autoclaved. I mean, a lot of these techniques, it's not something that it's so trivial that you just don't tend to put it in the methods. You tend to put more like sort of this is the technique, so you're inoculated, but you don't say exactly how you inoculated. As long as it's quality controlled and you can see the reliability of the data and it's been done multiple times, then that's all just required, and that's all that science really needs for it to be good science. You don't have to do it in a specific way that everyone tells you to do it, as long as you do it in a way that is repeatable, that you can do in your lab, and there's a certain style of technique. So if you're inoculating something, there's only so many ways you can inoculate it, and it's hard to go wrong. And as long as you've got a quality control there to say that you're doing it the right way every time, and the control strain, if you compare the control strains with the strains that you're testing, as long as you've got that difference and you've tested these things, then it should be a good result and a good answer. In fact, I would even say that sometimes if you yourself are fastidious and you just use the perfect technique, then you might find answers where there shouldn't be any answers at all. So yeah, that's what
I'm picking several things from your answer. One is the fact that there's already a culture of DIY already embedded in this type of research. Two, it's more of skill than anything. It's more of skill than anything, I suppose. And three, you can always compare with the kinds of results you get. The kinds of results and maybe just the general standards that are already there. But I think it's an important question, considering how instrumentation also plays a key role in the kinds of results that people can get from their work.
Definitely does. I think actually in the talk that paused, there was also a part in automation. So there are new kind of labs, and there's also on Jogel, there's lots of people that now no longer doing it manually, but they use robots that they've programmed to use these, especially with MIC testing that I talked about with the 96-well plates. The robots are really well designed for that. And on just one Jan lab on Jogel last week, well, actually the week before last week, we had a community call with someone who was creating robots for very, very cheap that could use the multi-peta devices, which is very, very interesting. We just have a question from.
Okay.
Yeah, Gamali, I was also just going to add that, my work in enzyme production was leveraging some microbiology techniques, and at some point we were not having access to like really high throughput equipment like the standard incubators, so we needed to build like a DIY version, which was like a thermoflux with controlled electronic panels just to control the temperature fluctuations that we needed and give the optimum temperature. It wasn't near to perfect, though we were able to get the temperature. I mean, it was able to hold it at some point, but over a period of time, we didn't get the kind of, say, 37 degrees temperature we wanted. At some point, we were having lower, not too lower than expected. But then at the end of the day, we're still seeing our bacteria growing perfectly well. Prior to that, one of the things when we were not having, we just decided to incubate the plates on the bench. We tried as much as possible to implement really stringent aseptic technique by using alcohol swabs and all of that. And we had really comparable results to what we were going to get in maybe a commercial, so to say, incubator. Subsequently, we moved there into building a DIY sterile chamber, which we tried to see how what the contaminating ability would be within the space, which also did work very well. The only thing that we saw that was missing from, say, a biological safety cabinet was that there was a laminar flow, which we tried to see if we could implement, which we were not able to do. That notwithstanding, it was able to provide that level of sterility for such a kind of work. And we haven't been using, since then, we decided to put it on hold and just use the flame method for our aseptic technique, which did work well. And it was out of those results that we did publish some work out of. So I think to look at the DIY approach, the safety quality, I think also boils down to the quality system of the implementing lab. Even if you are doing a low cost approach, it is still behoves on the implementing labs to just follow strictly the safety protocols and aseptic techniques. And at the end of the day, the result will be as comparable to maybe the standard equipment, and also just to mention, yeah.
Lovely, thank you very much for your follow-up response, Harry.
Yeah, thanks.
I just had a few questions in the chat. So Kichana asked about technical area of technical uncertainty and intermediate. So ATU versus intermediate, if there's any difference. And I think that there is no difference. In the end, these are just terminologies that people have made up to say they're uncertain. It's sort of an intermediate resistance, and I think it sort of depends on also the subpopulations. So if there's a colony inside, if they decide to report that or not, and that depends on the lab. I don't like these. So.
Chris, I think there's a question from Joseph. I'll just go to my chat to pick it out, okay. Which is saying that do we have to place the inoculated molar hinting plate with the 0.5 McFarland solution in the incubator for some time, say a few minutes before the application of the antibiotic? I don't know if you want to pick on this. I'm happy to.
No, anyway. I can't think of a good theoretical reason to, apart from the fact the discs might not stick to the plate. So just to ensure that the plate isn't too wet or too dry. You don't need to incubate it at all. There's no reason to incubate it. You can just put the discs on straight away. Yeah. Use the known. So right, for quality control strains, you say is it possible to use known organisms that you might have found if the commercial strains aren't available? And John was going to talk a bit about this, but yeah, you can. So I would normally use this for preliminary testing. And so if you wrote a paper about it, you would have to say how you got the strain, and your reviewers wouldn't be very happy with the fact that, in fact, your strain hasn't been characterized, and I'd probably have to repeat the results afterwards with a true quality control strain, I think. Yeah. And I think that's it. Is there any more questions? We've made a good 2 hours today, so brilliant. Thanks to everyone who's attending. Next week, we're going to talk with Gamali. And then the week after, we're going to have a talk with Harry. And then a few weeks after that, we're going to have practical sessions. And next week's talk will be about sort of AMR, the stewardship. So I think I talked about it before. But I'm certainly super interested to see what it said. So you can see this next week at the same time. Should we leave it there?
Thank you all, and looking forward to next week. I think we should probably do another group photograph one of these days, maybe next week.
Maybe in the last one as well. That'd be really nice.
Yeah.
Wow. We should wait for the last one to have another group photograph. Yeah.
Thanks, everyone.
Please feel free. If you have any other questions, you can ask on our Jugo platform. It's an interactive platform. Also, so
yes.
feel free to ask.
I think that just for just so that we kind of get used to the space that we created on Jogo.